Hey folks, I'm going to try to get through this and talk as fast as I can. Of course, you can always stop and pause it. You don't have to actually do this in real time. So, as promised in class today, this video is we're going to talk about some of the stuff that we've already talked about. So this is not going to be the first time you've heard it. It's called the sun's surface and the way scientists work. So we're going to combine two things into one presentation. All right, so we've already talked a lot about this. We talked about the core, the radiative zone, and the convective zone. All right, but sun also has an atmosphere, and the atmosphere has three parts. It's called the photosphere, the chromosphere, and the corona. So you can take a look at that on the diagram. So look at the first part, the photosphere. It's the part that we see. When we look at the sun, if you can look at the sun without hurting your eyes, it's the part that we see. It's a very thin layer. It's the sun is about like almost 100,000 kilometers in diameter, and this photosphere is only about 100 kilometers thick, so it's a very thin part, and it provides the light from the sun that we see. If you look at the sun with a special telescope, you will see darker spots on the sun. Those are called sunspots, and it's areas of cooler temperature. Just above the photosphere is something called the chromosphere. By the way, chromo means color. So through a special telescope with a certain filter, the chromosphere the sun will appear kind of reddish. Okay, this layer is only visible during a solar eclipse, or like I said, with a special filter that kind of recreates a solar eclipse. And finally, you have the corona, and the corona is not so much a layer, but it's this almost like this ejection of gases coming out of the sun. As you can see, all around this dark spot, this is a photograph of a solar eclipse. And all these this white cloudy areas, that's the corona, and it's very, 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 very not dense. About 2 million times less dense than our atmosphere. It's very thin, so to speak. Temperature is about 1.8 million. Scientists really aren't sure how the corona is hotter than the layer just below it, because usually as you go towards the center of the sun, it gets hotter, but it turns out the corona is about 1.8 million Fahrenheit, whereas the chromosphere is much, much cooler than that. The solar winds also originate here, and we may talk about solar winds just a little bit later in class. Okay, let's switch gears real fast to the stuff we talked about in class today. Talking about scientific method and how do scientists do experiments. The reason scientists ask questions is to get answers, obviously, and they want to know the who, the what, the where, the when about something. They're curious about something, so they do an experiment. So when you ask a question, you got to refine your question to where it's just asking about one thing only, and you take out all other language that's not important. Okay, today in class I had some of you take out words like you. What do you think will happen? Okay, you don't need that. So refine the question down to where it's very specific. All right, moving along. The next step to the scientific method is to try to predict what you think is going to happen. This is called a hypothesis. It's what you think is going to happen when you conduct your experiment, and it's based upon a little bit of research. So you don't go into the hypothesis to experiment blindly. You've at least looked up any kind of relative information about this. So it's the prediction of your answer or problem, and they're not guesses. No matter what you've been taught before, we don't want to guess at a hypothesis. Okay, if we have to guess, then we haven't done enough research. So the one we used in class today, for those classes that I had, third period, you didn't get to hear this. Coffee grounds will help plants grow. That was my hypothesis. Okay. What's next? Well, the next step is you've got your hypothesis. It's time to test it. And a test of a hypothesis is called an experiment. Now, you've heard of an experiment. You may have done an experiment. An experiment usually has two parts: the materials that you've used and the procedures you're going to do. It's kind of like a recipe. What were the ingredients you used, and what did you do with those ingredients? Okay? So the definition of an experiment is a test of a hypothesis. An experiment can only test one thing at a time. So you couldn't test, for example, do coffee grounds help plants grow, and what kind of coffee grounds are best for plants growth. You can't do both those at one time. That's two separate experiments. You can only test one thing at a time. Next, in an experiment, you're always going to have three important variables. You're going to have the independent variable, and that's the thing you're actually testing. In this case, you're testing whether or not coffee makes a difference. So coffee becomes your independent variable in this experiment. You have the dependent variable. This is what you're going to measure to see whether or not the coffee grounds made a difference. So the dependent variable is always measured to see whether or not the independent variable made a difference. Little hint here, the dependent variable is usually the data you're going to collect. Then finally, we have things like control variables. We're going to make sure that everything that we're not purposely testing, in this case the coffee grounds, are controlled. They're the same. For example, you don't want to use different plants in your experiment. You don't want to use different brands of coffee. You don't want to use different amounts of coffee. You want to use everything the same, except some plants will get coffee and some won't. Those are called control variables. As the experiment is running, or at the end of the experiment, you will collect data, information about the experiment. And again, this is usually your dependent variable. You will collect this data. You might organize it into a table, a chart, or a graph. And then you'll take a good hard look at it, okay? And that good hard look is going to lead you up into the last step. There's two kinds of data. Quantitative, which is kind of numerical. It involves the collection of numbers, the measurements, okay? For example, I said plants grew 14 inches, or another example, the mass dropped 40 grams. In either case, that's numerical. Qualitative are descriptive words. These are adjectives usually. Example, the food tasted bitter. The solution turned blue. Those are examples of qualitative or descriptive data. Most of the time in science, you usually get quantitative data. You'll get a bunch of numbers. Finally, you got your data. You looked at your data. Now it's time to ask that all-important question, does the data support my hypothesis? If you said that coffee grounds would help plants grow, and the plants that received the coffee did indeed grow better than the plants that did not receive the coffee, then yes, the data supported your hypothesis, and you're done. If it didn't, if your data doesn't support your hypothesis, then you got a choice to make. Are you going to give up in the experiment, in trying to solve your problem, or are you going to go back to step 2, make a new hypothesis, and do a new experiment? That's it. Okay, be sure you take notes. At least do screenshots, but notes are better. See you tomorrow.